Welcome to part one of our 13-part mass spectrometry training series. My name is Rod Chalk. I'm head of mass spectrometry at the Centre for Medicines Discovery, and today we'll be talking about fundamentals of mass spectrometry. We will cover what is mass spectrometry, and then we'll look at how mass spectrometers work. We'll look at ion sources, we'll look at ion optics, we'll look at several different types of mass filter and different types of mass detector. So this is a typical workflow for mass spectrometry. There's a lot of work involved in sample preparation, and also a lot of work involved in data analysis. The mass spec part of any experiment is relatively fast in comparison, and I've drawn the mass spectrometer here as a black box, and it is perfectly acceptable to accept a black box approach and not worry too much about what is actually happening inside the mass spectrometer, and you can focus on getting your sample preparation correct and doing your data analysis. However, if you're a scientist, you will naturally be curious about what goes on inside the mass spectrometer, and I would say the more you understand about this, the stronger position you will be to use mass spectrometry as a tool. So what is mass spectrometry? In essence, we are generating charged particles, ions, and we are generating an ion beam. And that ion beam we can manipulate under high vacuum. We can measure the intensity of those ions, but crucially we can also measure their mass to charge ratio. Mass spectrometers do not measure mass. That may seem counterintuitive, but it's true. They measure the ratio of mass to charge. In addition, we can measure parent ions, which can then be fragmented. And the daughter ions, they can also be measured in terms of their mass to charge ratio. So that's in a nutshell what it is. How do mass spectrometers work? Well, mass spectrometers are generally quite complicated instruments, but the easiest way to understand how they work is to go right back to the origins of mass spectrometry and have a look at a very, very simple instrument. And the one I've chosen to show you today is this one. This was invented by J.J. Thomson in 1913, and it's very simple. It consists of a partially evacuated tube. It's actually filled with gas, that is the analyte, and here we have a cathode with a voltage applied to it. This is our ion source. That ion beam will pass through these slits and then be collimated, and then it can pass through two types of mass filter. These magnetic coils will generate a magnetic field, so it can be bent in that magnetic field. These electrical deflection plates will generate an electrical field, and it can be bent in the electrical field. And then the detector is simply a fluorescent coating on the inner surface of this partially evacuated tube. And when an electron beam or a particle beam, a charged particle beam hits this fluorescent coating, it will fluoresce and you'll be able to see it. Very simple instrument, simply consisting of an ion source, two different types of mass filter, and a detector. And when you look at different types of mass spectrometer, and we will look at different types of mass spectrometer in the course of this presentation, I want you to think in terms of these three parts. Every instrument has to have an ion source. There are different types. Every instrument has to have a mass filter. There are different types. We will discuss them. And every instrument has to have a detector. So this is a schematic of how this instrument works. You can see the ion beam being bent by these electrodes. And although it's very simple, this instrument was very powerful in that Thomson was able to use it to separate isotopes of neon. So he was able to separate neon 20 from the heavy isotope neon 22. And he also correctly identified this species, MZ11, as the doubly charged particle of the neon 22. So before we look at different types of mass spectrometer, let's think about what we need in order to make a good mass spectrometer. What do we need to make a mass spectrometer powerful? Well, I guess the most important thing is mass accuracy. The more accurate your instrument is, the better. Typically, mass accuracy is quoted in parts per million, or sometimes in parts per billion. Related to mass accuracy is resolution. These two things, mass accuracy and resolution, generally go hand in hand. Resolution is the ability to separate peaks. And the more peaks you can separate in your mass spectrometer, the greater the resolution and the greater the information content. Sensitivity is obviously going to be very important. The more sensitive your instrument is, the better. But related to sensitivity is the issue of dynamic range. It's particularly important in biological samples because we have species of interest which may be at very low abundance, mixed in with other species at extremely high abundance. So can your instrument operate over a very high dynamic range? Fragmentation is important. Fragmentation is the ability to take parent ions, fragment them and measure the fragments, and this equates with information content. So the better your instrument is at fragmentation, the more information you will get. Scan speed. So this is the ability to generate spectra and look for fragments. And the faster you can scan, the greater the sample complexity you can deal with. And finally, you will need your instrument to operate over a suitable mass range. So I've indicated here we can operate with small molecules, peptides and oligonucleotides, proteins, whole proteins, multi-protein complexes, and finally macromolecular assemblies. And you may need your instrument to operate over this entire mass range. Let's move on now to the 1960s and look at a more sophisticated instrument. This is the type of instrument that was in textbooks when I was at school and university. And again, it consists of three components. We have an ion source generating an ion beam. We have a mass filter here. In this case, it's a very large electromagnet. And you can see that that ion beam, as it passes into the magnetic field, it's going to be bent. And it's not hard to work out that the lighter ions will be bent further in this magnetic field, and the heavier ions will be bent less in this magnetic field, and they will be separated in terms of their mass. And then we have some simple detectors here, Faraday cup detectors, an array of these in order to generate our spectrum. I now want to mention something about ion sources. I have listed here 7 different types of ion sources: spark ionization, chemical ionization, APCI, thermospray, fast atom bombardment, electrospray, and MALDI. And I've given the various dates of when these ionization techniques came into use. I have labeled these ones in red. The reason for that is I don't really know very much about these ionization techniques, and the reason for that is because I'm a biologist by training, and these I've labeled in red are hard ionization techniques. This means that for a biomolecule like a protein, they will just cause it to disrupt the structure. And these techniques are of no use in general in biology. However, these 2 ionization techniques, electrospray and MALDI, these are soft ionization methods. And the invention of these 2 techniques was responsible for a huge transformation in mass spectrometry and a huge explosion in the use of mass spectrometry for use in biology. So electrospray was invented by John Fenn. MALDI was co-invented by Koichi Tanaka. And in recognition of the enormous contribution that these two ionization techniques have made in the field of biology, Tanaka and Fenn were jointly awarded the Nobel Prize in 2002. Let's look at a more sophisticated instrument now. This is a MALDI Topoff mass spectrometer. MALDI Topoff stands for Matrix Assisted Laser Desorption Ionization Time of Flight. So MALDI is the ion source and Topoff is the mass filter. And it's not difficult to work out how a Topoff tube works. We accelerate our ion in electrical field and then it will drift down the time of flight tube until it hits the detector, and we will measure the time it takes for that to happen. We can measure time extremely accurately electronically, and then we can use that to derive the mass to charge. So the drift tube, the drift region is here in our schematic, and in the real picture the time of flight tube would be in this orientation, horizontal. So we need a high vacuum. There's a high vacuum inside this chamber. It's 10 to the minus 7 torr. 1 torr is 1 bar, approximately 1 bar of mercury. And we need to have a sample. Now in MALDI, the sample, liquid sample, is mixed with a liquid matrix. And then in small volume, so this is about 2 microliter volume here on a metal sample target, and it's allowed to crystallize, and so the matrix and the sample co-crystallize on this metal target plate. We now need to introduce the metal target into the high vacuum, and we do this through a sample loading chamber. And this works rather like an airlock in spacecraft, except it's the opposite way around. We're moving from atmospheric pressure to high vacuum, and this is done mechanically, and it will load the sample into the source chamber. This is our sample now sitting in the source chamber, and we have a video camera. We need a video camera because we're going to fire a laser onto our sample spot. You can see that there are multiple sample spots on the target, and we need to, what we'll do is we don't steer the laser onto the target, we steer the target onto the laser, but we need a video camera in order to do that. This is our laser. It's a 337 nitrogen laser, and there's a laser attenuator and a prism to bend that laser beam so that it will now land on our target plate. And when it does that, we will have ablation of the sample. So it will be ablated into the vacuum. The matrix becomes charged, and then there is a charge transfer process which transfers a single charge from one of the matrix ions onto an analyte, and we end up with singly charged analyte ions in MALDI. When we have that plume of ions, we then apply a voltage, and that voltage is applied from this voltage grid onto the ground grid, and that is typically 15,000 volts. So these ions are given a big kick. They will pass through this collision cell. There's an optional collision cell, and then they will pass into the flight tube. In the flight tube, this is a drift region, so there's no high voltage applied. Your ions are being accelerated and they drift down this flight tube. As they do that, they will tend to, so all the ions have the same charge. They can either be all positively charged, or you can reverse the polarity, they can all be negatively charged, but they will tend to want to repel each other. There is Coulomb repulsion between these ion species, and to counteract that repulsion, the function of this beam guide wire is to pull the ions closer into the center of the tube. They will then pass through a timed ion selector. So this is a type of mass filter. So we can select an individual ion of interest, and then finally they will hit the linear detector here. So we can measure the time of flight. We now have an electrostatic mirror. So this is an electrical field applied to the opposite charge, so the ions will slow down and then eventually stop inside this electrostatic mirror, and then they're reflected back onto a secondary reflected detector here. And so again, think in terms of ion source, mass filter, and detector. The MALDI source chamber is your ion source. The flight tube is the first mass filter. The timed ion selector is the second mass filter. We have the first linear detector. We have the reflector, the electrostatic mirror, as another mass filter. And then finally, we have a second detector. So when MALDI was first developed, it didn't make very much impact. And the reason it didn't is because the resolution of the technique was extremely low. Typically, MALDI resolution was a few hundred, not that much better than gel. However, that changed in the late 1980s with the development of delayed extraction. And the problem with MALDI is when your laser hits the target and your ions are ablated, if we imagine that we have ions here, all of the same mass, in that cloud of energy, some of these ions will have a high velocity and some will have a relatively low velocity. And the problem is when they drift down the flight tube and hit the detector, they're going to arrive at different times. They're going to be out of focus. So the solution to this turned out to delay the high voltage pulse. So what happens is when your laser hits the target, there is no voltage applied, and you wait for 150 nanoseconds, and then you apply the voltage. And what happens is in that 150 nanoseconds, the fast-moving ions have penetrated a long way into the acceleration field, and the slower-moving ions have only penetrated a short way into the accelerating field. When you then switch that acceleration field on, the ion that has traveled a long way through the field is accelerated by a small amount, but the ions that have traveled a short distance into the field are accelerated by a large amount. So as these ions drift down the flight tube, the faster ions, the slower ions will catch up with the faster ions, and they will all reach the detector in focus at the same time. So this made an enormous difference. This improved the resolution of these instruments from a few hundred to several thousand. So the function of delayed extraction is that the ions of the same mass are focused in time, and this achieves improved resolution, improved mass accuracy, and improved sensitivity. I mentioned the Reflectron, the ion mirror. And the idea behind this is also that ions of the same mass are focused in time. It also allows the Topoff drift time to be increased, and that results in improved resolution, improved mass accuracy, and improved sensitivity. And how the ion mirror works is very simple. If we imagine again, we have two ions of the same mass, but traveling at different velocities. So the red ion is traveling slightly faster than the blue ion. As they travel down the drift tube and enter the Reflectron, the faster moving red ion has more kinetic energy, and that will pass further into the Reflectron before it slows down, it stops, and then it reverses and accelerates back out. The lighter blue ion has less kinetic energy, so it's not able to penetrate as far, so it will only come to this distance, and then it's accelerated out again. So as these ions leave the Reflectron and reach the detector, they will reach the detector at the same time, so they're focused in time. So a Reflectron makes a big difference to the resolution of the instrument. It increases the effective path length of the flight tube by about 3-fold. And originally reflectrons were rather expensive. Now every TOF instrument comes with a reflectron. This is another type of moldy instrument. This is a moldy TOF. So this is one of the first instruments we'll look at where it's the instrument is capable of fragmentation. So like the other moldy instruments I've shown you, we have a laser. The laser generates our ion beam, and that is accelerated in the first time of flight region. It will then pass through a mass gate that allows us to select parents for fragmentation. The selected ion is fragmented in the collision cell, and then as it leaves the collision cell, those fragments undergo a second TOF acceleration before reaching the detector. That's a moldy TOF. And another thing that made an enormous difference to time of flight instruments was the development of orthogonal TOF. And the idea behind orthogonal TOF is that you have a dispersity of ions when they're being accelerated in this plane. And the idea is to collimate them into a flat beam. So this flat beam has zero velocity in this plane at 90 degrees accelerating voltage. And the idea is that if you applied accelerating voltage at 90 degrees to this beam, then everything will have zero velocity in the plane of acceleration. So you have a pusher here, you accelerate at 90 degrees to the ion path, and that again will give you improved resolution and mass accuracy. So that's a time of flight mass filter. Let's look now at a different type of mass filter. This is a quadrupole mass filter. So as the name implies, a quadrupole has four rod electrodes, and we apply a radio frequency to these electrodes. So there is a polarity switch between the electrodes at RF frequency, and what happens is as different ions, different masses pass between these electrodes, some ions will be accelerated towards one rod, others will be accelerated to the other. But you can see this purple ion, this is at the resonant frequency of the rods. So this is at the radio frequency, is the same as the resonant frequency of that ion, and it allows it to pass through the quadrupole filter. So what we can do is we can scan across frequencies, and when we scan, that will generate a spectrum. And the resolution of quadrupole mass filters is not high. The resolution is dependent on unit mass dependent. But the quadrupole is very good for selecting parent ions for fragmentation. So that's a quadrupole mass filter, and they can be used in this instrument. This is called a triple quad. As it implies, it has 3 quadrupoles. So we have an ionization source here, and we have the first quadrupole mass filter. This allows us to select a parent ion for fragmentation. We have a second quadrupole, which is the collision cell. This is where the parent ion will be fragmented. And then the third quadrupole allows us to select the mass of the fragment ions. And then finally, those fragment ions will reach the particle multiplier, which is the detector. So again, this is our ion source, these are our mass filters, and this is our detector. So that's a triple quad. Triple quad is used largely for quantitative work in drug metabolism and pharmacokinetics in the pharmaceutical industry. So another thing that made a big difference to mass spectrometry of biological samples was the invention of the cutoff mass spectrometer. This was done originally by Micromass, now Waters, in 1996, and they had the idea to couple together a quadrupole and a time of flight filter, mass filter, in the same instrument. And I want to now show you a video. This is an agilent cutoff mass spectrometer, and we're going to open the source chamber. And you can see here, this is the sample nebulizer. So we have a stream of charged droplets emerging from the sample nebulizer in electrospray. And alongside, you can see the reference nebulizer, that is for internal mass correction. And these charged particles are drawn into the source of the mass spectrometer. And as they enter the source, they will encounter a stream of hot gas, and they are desolvated, so that the water, the solvent is lost, and they become charged. The grey coloured particles, these are neutrals. We don't want those. All the charged particles you see have the same charge. They're all positively charged. They're passing now into the ion guide and through a couple of Einzel lenses, and they're now entering the first quadrupole. So in the quadrupole, we have mass gating. So we're gating just the green ions, and then they will pass through another lens into the collision cell. Here in the collision cell, they collide with an inert gas at high velocity, and that will cause the ions to fragment into smaller particles. And you can see these particles now passing through into a second octopole ion guide. Now remember, this is orthogonal Topoff. So what we want to do is we need now to flatten that beam in the horizontal plane. So pass through this slit through a second octopole ion guide, quadrupole ion guide, and pass it through the slicer. Again, we're removing these ions so that everything has zero velocity in the vertical plane. And now we've entered the Topoff chamber. So they receive an acceleration, ions receive an acceleration, and they're now drifting in the flight tube. This is Reflectron. The ions stop and then reverse back down onto the detector at the bottom of the flight tube. And here they strike a microchannel plate detector. The signal is converted to electrons and back into photons, back into electrons in the photomultiplier tube, and then that will generate our MZ spectrum. I'm going to return to our presentation. And let's now look at a different type of mass filter. This is the ion trap mass filter. Now, if you understood what was happening with the quadrupole, if you take a ring electrode and you apply an RF frequency to that ring electrode, and then you add an end cap, two end cap electrodes either side of that ring, you can use RF frequency scanning to RF frequency to trap the resonant frequent ions in this space within the ion trap. And we can scan through frequencies, and we sequentially eject those ions onto a detector, and that will generate a spectrum for us. So this is our ion trap mass filter. This is an example of an ion trap instrument. Remember, we have the different components. We have electrospray source, we have the trap, that is the mass filter, and then we have a detector here. So that's ion trap. This instrument I want to show you is an FTICR mass spectrometer. So this is a very large instrument, very expensive instrument. It's the instrument that will generate the highest resolution. And FTICR stands for Fourier transform ion cyclotron resonance. And the reason these instruments are very large and very expensive is because of this. This is a superconducting magnet, and this is necessary to generate a large magnetic field, a very powerful magnetic field in which you generate a cyclotron. And these instruments are expensive because you basically have to design your lab around this, and it's quite expensive to maintain. So this is a schematic of the FTICR. This is your ion source, this series of ion guides, and packets of ions are sent into the ion cyclotron resonance cell, which is within this. And here, this is 9.4 Tesla superconducting magnet. And I can show you a video of how this FTICR works. So this is our FTICR mass spectrometer, and we are going to generate an ion beam that passes through a series of quadrupoles, and then we want to generate an ion packet. And this ion packet is then sent down into the ion cyclotron region. This is the center of the cyclotron. We are going to apply an RF voltage to these electrodes, and that's going to induce the ions to cycle in the magnetic field. Now remember that we have a 9.4 Tesla magnetic field, and as charge moves through a magnetic field, it's going to generate a current. So that induces a current in these alternate electrodes. And that current will generate a complex signal waveform. That waveform needs to be so FTICR is a
very
transformation,
very
powerful
will
technique.
spectrum.
It was considered the gold standard in mass spectrometry. However, these instruments are very large, they're very expensive. Thermo were able to develop an instrument that could deliver much of what FTICR can deliver at very much reduced cost. And this is the mass filter called an Orbitrap mass filter. It's a mass filter and it's a detector at the same time. I didn't mention this, but the FTICR, that is a mass filter and a detector together. You can achieve something similar with an Orbitrap. But how this is done is the cycling happens here. So this is an ion trap, but it's an orbital ion trap, so it has this central spindle. And the C-trap here generates a packet of ions. These ions are sent into the Orbitrap, and then they are induced to spin around this spindle. And in doing so, they will generate a signal, which we can transform. I'm going to show you a video. So this is a diagram of an Orbitrap instrument. This is what it looks like in real life. This is your ion source. We have here a linear ion trap. This is the C-trap. This is our collision cell, and that is your Orbitrap. I'm just going to play this video and we can see how this works. So this is an ion beam in our Orbitrap mass spectrometer. It's now passing through an active beam guide. This allows us to remove neutrals. They're not bent in the guide. And the beam is now passing into a quadrupole mass filter. Here we can select the ions that we wish to fragment. Passing through the quadrupole filter, they then pass beyond the Orbitrap into this routing multipole. And here, this is where fragmentation takes place. and we can accumulate ions and then pass them into the C-trap. When they pass into the C-trap, they are then focused into a small packet, and then that packet then passes into the orbitrap. The ions are cycled in the orbitrap and they will generate a signal. Okay, so that's an orbitrap. Here's another type of TOF instrument. This is an ion mobility cutoff. So this has lots of the elements that we already described in a cutoff. We have electricity by ion source, we have quadrupole here, we have off acceleration, we have two ion mirrors, so we have a double reflectron here. But what's different about this instrument is this. This is an ion mobility separation cell. What happens here is we have an inert gas, and your ions collide with this inert gas at low velocity. So they're not fragmented. So as the ion stream passes through this cell and collides with the helium gas, the ions are retarded. And they're retarded proportionally to their collisional cross-sectional area. So if an ion is large and it has a large collisional cross-sectional area, it will be retarded relative to a smaller ions before it reaches the TOF mass filter. So this is ion mobility cutoff. There is another type of ion mobility, and this is trapped ion mobility. So in the cutoff I just showed you, we have temporal separation of ions based on their collisional cross-sectional area. And you can also separate them spatially. This is not normally used. In the trapped ion mobility, what happens is your ions are trapped in space and also trapped in time. This is what it looks like. So this is our trapped ion mobility cell here. This is a quadrupole, so we have an ion source here. This is our quadrupole. We have a Topoff acceleration region here. And so what happens is the ions are separated by virtue of their collisional cross-section. They are accumulated on that basis, and then they are sequentially released into the mass analyzer of the mass spectrometer. And the effect of that is to accumulate these ions into these small packets, which are sequentially, so they are accumulated and trapped and then eluted, and that can happen faster than they travel into the mass spectrometer. So we can separate, we can achieve increased resolving power, and also we can achieve increased sensitivity with trapped ion mobility. I now want to say something about fragmentation. There are different methods of fragmentation. The most commonly used is collision-induced dissociation, or CID, and the peptide bond in a protein or a peptide will fragment in a predictable way, and predominantly it generates these B and Y fragment ions, the B ion from the N-terminal fragment and the Y ion from the C-terminal fragment, and we refer to these as daughter ions, child ions. And this is what a fragmentation spectrum looks like. So this is a triplet peptide. This is the sequence of the peptide. This is the fragment spectrum. The B ions are labeled in red and the Y ions are labeled in blue. And we can derive the sequence from the separation of these ions. So in order to generate sequence data, you need to generate a nested series of fragments, similar to what you do in conventional DNA sequencing. But unlike DNA sequencing, we generate fragments simultaneously from both ends. This is why we get the B and Y ion series. This is a very nice fragmentation spectrum, quite a long sequence run. Another type of fragmentation is ETD, or electron transfer dissociation. Now this is achieved, fragmentation is achieved by addition into the ion stream of a negatively charged compound. So we have a source of this fluoranthane gas. This is negatively charged, and as it interacts with your ions, this negative charge induces cleavage of the peptide bond, and it cleaves in a slightly different way, generating these C fragments at the N terminus and Z fragments at the C terminus. ETD is in a way better than CID because the fragmentation is random. If you think about it, random fragmentation is what you're looking for. You don't want non-random fragmentation. And the MSMS coverage is more extensive. And another advantage of ETD is post-translational modifications, particularly phosphorylation, but also other post-translation modifications, are not lost. So they'll still be on the fragment ion. Here's a comparison of ETD versus CID fragmentation for the same peptide. And you can see that random fragmentation for ETD generates roughly the same intensity for the different ion species. Whereas CID fragmentation, you can see you've got some good signal here and then nothing. So you get, this is an example of non-random fragmentation. So our coverage for ETD is more extensive, and critically, our ion score, so this is how good the match is with the database, is much better. So here we have an ion score of 94 versus an ion score here of 56. Now finally, I just want to say something about detector types. The most common detector type in mass spectrometry is a microchannel plate detector. The way that this works is we have a high voltage applied across here, and as the charged species hits the detector, it will strike off. If it's traveling fast enough, it will strike off a secondary electron. That electron will then strike the plate here and generate more than one secondary electrons. And those secondary electrons in turn will generate more. My pointer here, they'll generate more secondary electrons, and you can see that you're generating an electron cascade. So that finally, at the end of this process, you'll generate sufficient electrons to generate a current, and that current can be measured. This is a simpler detector. This is a Faraday cup. We have a charged species hitting the cup and generating a small signal. Fourier transform ion cyclotron we've already looked at. This is, you see how that works. Just for completeness, I've added this type of detector. This is a single particle detector, a superconducting tunneling junction detector. This is basically for research only. This is not commercially available. So that's all I want to say about the fundamentals of mass spectrometry. Just to summarize, we discuss what mass spectrometry is. An ion beam is manipulated under high vacuum, where the mass to charge ratio of ions is determined. Good mass spectrometer will, what makes a good mass spectrometer is accuracy, resolution, sensitivity, the ability to generate fragments, high scan speed, and a good mass range. We looked at different ion sources. We discussed hard ion sources versus soft ion sources. We specifically looked at MALDI and electrospray. We looked at different types of mass filters. So we looked at magnetic sector, time of flight, quadrupole, ion trap, Fourier transform ion cyclotron resonance, and Orbitrap detectors. And these are different types of ion optics, and these are just different ways of focusing ions. We looked at the Topoff instrument. We looked at different types of fragmentation, CID and ETD, and then finally we looked at different types of detectors. Now I hope you've understood something about mass spectrometry from what I've said. However, if you've understood nothing, I would say, well, possibly it really doesn't matter, because the real power of mass spectrometry does not depend on knowing how instruments work. The real power of it is understanding what these instruments can do. And that's what I'm going to be talking about in the remainder of these training lectures. And I just want to finish it off by mentioning this man. This is Professor Jules Hofmann from CNRS in Strasbourg. When I was a young researcher, he was my principal competitor. And Jules started using mass spectrometry in 1992. I didn't start using mass spectrometry until 1996. I just want to say that Jules Hofmann won the Nobel Prize for Physiology and Medicine in 2011. And yes, learn from that, please. And just want to say next week we'll be talking about protein analysis using tandem mass spectrometry, and I'll be very happy to answer your questions now.